जी स्टूडेंट्स अस्सलाम वालेकुम। आज हम पढ़ेंगे रिगार्डिंग ट्रांसडक्शन एंड ट्रांसडक्शन अ बैक्टीरिया phage transfers DNA from a donor cell to a recipient cell. There are two types of transduction. Number one is the generalized transduction and number two is the specialized transduction. Generalized transduction, a random fragment of the bacterial DNA is packaged in the phage head in place of the phage DNA. Normal excision is done in this process. In the second process, the second is the specialized transduction. The combination between the phage chromosome and the host chromosome produces a phage chromosome containing a piece of bacterial DNA. In the generalized transduction, phages are of two types: virulent, and they multiply and lyse, and temperate, they has a choice, they can enter into the lytic or the lysogenic cycle. The generalized transduction can be done in virulent and temperate virus. Generalized transduction particles are generated during the lytic cycle of these phages. Not all bacteria mediate transductions. In some bacteria like T1, T246, the virus degrade the host genome. When the virus degrades the host genome, it utilizes the nucleotide for the synthesis of the phage DNA. So host genome is not available for packaging. This is done in some bacteria, mostly in some T1 bacteria. As I already told you that in some viruses, transduction cannot be mediated. These are those viruses in which the genome is completely degraded. From the generalized transduction phages, E. coli phage P1, Salmonella phage P22, and Bacillus subtilis phage PBS1 and S10 are extensively used for the transduction. Normal excision of the phage lambda, normal excision of the lambda prophage. In the generalized transductions, phages can transport any bacterial genome from one cell to another. Thus, the name generalized transduction is given to this process. In the phage lambda, in the lambda prophage, loops out and ATTBP pairs with the ATTPB. This is a site-specific recombination in the uh, generalized transduction. If you look here, in the generalized mechanism, in the generalized transduction, the prophage loops out, ATTBP binds with the ATTP, and after that, site-specific recombination occurs, and ATTBP and ATTPB excise the phage lambda chromosome. So this process is the normal excision. Sometimes, during the generalized transduction, a random fragment of the bacterial DNA is packaged in the phage head in the place of phage DNA during the process of normal excision. Anomalous excision of the phage lambda phage, when sometimes the prophage loops out anomalously with ATTBP and ATTPB, and at that time ATTB and ATTBP are not paired properly. During this improper pairing, recombination excises a lambda chromosome carrying E. coli gene. Say, for example, if you look in this diagram, uh, when the phage lambda recombines with the bacterial DNA, when the phage lambda recombines with the DNA, say, for example, on its one side is a gal gene, on its other side any biosynthetic gene. So when second recombination takes place, this viral genome is excised from the bacterial genome. This virus genome is excised from a bacterial genome. Okay, and After this anomalous recombination, the bacterial gene, the bacterial gene is inserted into the viral genome. Here you can see the gal gene is inserted here because after the gal gene recombination take place, and the bacterial gene, say for example, J remains in the bacterial genome, remains in the bacterial genome. So there is the exchange of genes takes place. In the specialized transduction, this is a characteristics of viruses that transfer only certain genes between bacteria. Bacteriophage lambda is the best known specialized transduction phase, which carries only the gal gene. In this process, integration of the lambda D gal positive at the ATTB produces a gal positive gal negative partial diploidy condition in the bacteria. Say for example, when this lambda D gal containing virus will attack on the bacterial genome, there will be again a site-specific recombination between the viral ATT and bacterial ATT site. Say for example, this bacteria is gal negative containing gene, and when the virus infect this gal negative recipient, there will be a partial diploidy for gal gene. So it will be gal positive and the gal negative genes in the bacterial cells. So this is the partial diploidy condition. When this partial diploidy condition will establish during this condition, a double crossover will be take place between these two genes, between the gall positive and the gall negative genes. Why when this double crossover takes place in the bacteria, the the complete virus genome is not inserted into the bacterial genome. Only the Gall negative and Gall positive genes will be exchanged. So we can say that Gall positive gene will be inserted into the bacterial genome and Gall negative gene will be inserted into the viral genome. So there is the exchange of genetic material in this specialized transduction. So up till now, there are three parasexual processes: transformation, conjugation, and transduction, which are occurring in the bacteria. These processes can be distinguished by two criteria: whether the gene's transfer is inhibited by deoxyribonucleases and whether it requires cell contact. Next is the evolutionary significance of genetic exchange in the bacteria. Genetic exchange is an important process in the bacteria as it is in other organisms. Mutation is the source of new genetic variation. Mutation is the source of new genetic variation, mostly because of the UV radiation. Because, say, for example, the bacteria become mutated. Recombination produces new combination of alleles. Um, because of the recombination, new alleles are introduced into the population. So mutation is very important factor to introduce the new alleles in the population. Transformation, conjugation, and transduction generate new combination of the genes in bacteria to allow bacteria to adopt a new environment. So because of this process, the bacteria can adopt a new environment for their survival. This is all about for today. Well, and hoping it will be easy for you to understand. Tomorrow we will learn more about some other processes involved in the molecular genetics. Thank you. Good luck.